Okay, so now gets a little bit more complicated. I showed you one example of how complete dominance happens, one of how codominance happens, and now it's incomplete dominance. There are many ways that this could come about. I will show you some examples. So it can either be from not having enough units of an enzyme to do the job it needs to do, so the job gets partly done. It could be that the gene for the enzyme is not mutated, but there is a mutation in the promoter sequence, which then says not as many units of that enzyme gets made. So your mutations that alter your phenotype would be a mutation on the gene itself. So maybe you have one version of the enzyme that it works, one that doesn't work, and if there's haploid insufficiency, then you're only going to get as much job done as the functional enzyme. Or the mutation could be on the promoter, which maybe both alleles of that gene are the functional enzyme, but if there's a change on the promoter, then you don't make as many mRNAs of that molecule, which may not have produced as many proteins after translation. And the third scenario, which is still on the gene expression of that gene, maybe the structure of the gene or the sequence of the gene is the one that works, and the promoter is the one that allows the RNA polymerase and the transcription factors to bind. But if there is a mutation on the transcription factors, so a mutation on another gene, then that affects the phenotype of this one gene. So you'll see examples of that. So example of haploid insufficiency. Classical example is Snapdragons, and in reality it's not this simple. We'll see some pictures of this. You have a red flower, which is red because it makes this pigment that's called anthocyanin. So if you have a bunch of anthocyanin, the flower is red. And if you don't have a bunch of anthocyanin, then it's white. So we're going to say that the white is the background color, just like the yellow was the background color in here. So remember, in this case, the endosperm is yellow, and then the cotyledon, which is the wrapping paper of the seed, if it's see-through, your seed is yellow. If the cotyledon is green, the seed is green. So in this case, you're going to say that the white is the absence of the red pigment. So how do you make this red pigment? There are many steps, but I'm going to focus on one. There is this enzyme called Chalcone Synthase. So it's some enzyme that works on the pathway of making anthocyanin. And this enzyme is dimeric. So you will learn that the proteins, they can be monomeric. So there is one gene that gets transcribed, it folds, that's the protein. Other ones, you need subunits to get together for it to work. So this dimeric means that the enzyme has two subunits. Okay, then we're going to look at two different alleles. The allele that encodes red and the allele that encodes white. So the allele that encodes the Chalcone synthase for the red allele, this enzyme works, or the subunit is a functional one. Then on white, the Chalcone synthase subunit doesn't work. So here you have both enzymes work, you make something red. Here, both of the enzymes don't work, so you don't make anthocyanin, then it's white. And then in here, an enzyme works and then another enzyme doesn't work, so you are halfway. So how does that work? And you could think about it as a monomeric enzyme, as in not as many proteins made, But in this case, there's another wrinkle, that is the fact that the enzyme has 2 subunits. So here's the picture of the Chalcone Synthase. And here you'd have, let's say that the yellow one is the one that works. So you have the functional subunit of Chalcone Synthase here, and here's the other functional unit. It makes one enzyme that works well. And here you have a white subunit and a white subunit, meaning this one is the one that has the enzyme doesn't work, this one doesn't work. So 2 parts that don't work, they don't work at all. So that is that, and then that is that. And in here, you have the 2 alleles that if they come together, now you make an enzyme that sort of works, sort of doesn't. So it's not always like 100%, 0, or 50%. And you'll see examples of that in homework. But that's active, this is not active, and this just has decreased activity. So it could be that it's because of the enzyme activity, or it could be the fact that there's some transcription. Oh, actually, there's a later example. Let me focus on this one, sorry. So here I'm explaining or contrasting haploinsufficiency with haplosufficiency. The haploinsufficiency, you have alleles. So if you have only one version of the functional allele, which would be this one, it's not enough, that's a sufficient part, to produce the phenotype of when the allele is functional. And then in the case of haplosufficiency, having one allele is enough to make the same phenotype as the one when both alleles are functional. So in this example, here is your functional allele. It makes the dimeric, the 2 units, and makes a dimeric enzyme that works. And how is it that having one allele is enough? Okay, you have that one allele, but you have 2 mRNAs made out of it, or you could even have one mRNA that gets translated twice. And then the non-functional one doesn't actually even get transcribed or translated. So this subunit that is the non-functional doesn't actually mess with the activity of this, so it doesn't bind any of these subunits. So you don't get this chimeric enzyme, even though you only have one gene that encodes a functional enzyme. The fact that the second one doesn't get made, then this allele doesn't interfere with the action of this one enzyme. So this would be a case of haplosufficiency, and here I'm showing you the haploinsufficiency.